Hi everyone, myself Dr. Manjunath C. I'm an associate professor in the Department of Chemistry, RV College of Engineering, Bangalore. In this recording, I'll be discussing about the smart sensing material, which is also there in Chemistry of Smart Materials and Devices. And this course has been introduced for the first year engineering students for computer science and allied branches. So, this is the syllabus that we have in the Unit 4, that is Quantum Materials and Sensing. And in my earlier recordings, I have discussed about CNTs, graphene, and superconducting materials, etc., and also about polyaniline. So, in this recording, I'll be discussing about the electrochemical sensor, and also in another video, I'll be covering piezoelectric sensors. I just split the recordings into two about electrochemical sensors and piezoelectric sensors. So, let me start with electrochemical sensor. Before that, let us understand what is the meaning of sensor, first of all. Sensor is a device, okay, that measures, it's an electronic device that measures the change in physical properties like heat or light, sound or pressure, and not only physical properties, or sometimes also the chemical properties like pH of the solution, the smell, taste, concentration, humidity, etc. So these physical and chemical properties into the measurable parameters like like resistance or the voltage or the current. So these are the parameters of our interest in an environment and transforms that into electronic signal. So you can see that this representation of sensor, you can clearly understand that the various forms of energy like mechanical energy, thermal, electrical energy, or magnetic property, radiation properties, or chemical properties are being converted to some measurable electrical properties like voltage, current, charge, or sometimes phase change, amplitude, frequency, etc. So we have some device that is called sensor, which converts these physical and chemical properties into electrical properties. For example, with respect to the mechanical form of energy, the physical quantities are measured or length, area, volume expansion or displacement, the velocity, mass flow, or pressure, etc. So with respect to thermal, the temperature change or the specific heat of the material or entropic change or heat flow, specific capacity, etc. Electrical properties such as the change in the voltage, or change in the current, resistance, inductance, capacitance. So these are the properties. Similarly, magnetic properties like field intensity, the magnetic flux density, magnetic moment, permeability, etc. Similarly, with respect to the radiations like intensity, phase, wavelength, polarization, etc. And with respect to the chemical properties like the composition of the analyte and its concentration, the rate of the reaction, the variation of the pH, whether the analyte is undergoing oxidation or reduction. So these properties are converted to the measurable electrical properties. Such devices are called sensor devices. So in this recording, I'll be completely focusing on the electrochemical sensor. So electrochemical sensors convert chemical information. So the chemical information into electrical signal. Here the electrical signal generated are due to the redox reactions. So redox reactions means it is a reduction and oxidation. Oxidation means the release of electron. Reduction means the consumptions of the electrons. The output measured in this sensor device is sometimes we measure the current, or in some other cases we measure voltage or resistance. Based on that, we classify electrochemical sensors into amperometric, potentiometric, and also conductometric electrochemical sensors. So this technique, this sensor are widely used in chemical, biological sensors, gas sensor, and widely used in biomedical field. And those who are interested to develop a sensor as part of your experiential learning or project-based learning, you can just refer this article, which is recently published in ECS Sensor Plus. It's an open access research article, review article This is exclusively discussed on electrochemical sensors and food. So they have covered both conductometric, potentiometric, amperometric, and impedometric types of sensors. So this will help you for your project-based learning. Let us understand the working and the construction of this electrochemical sensor. So electrochemical sensor basically it consists of three electrodes. So this is the working electrode. It is also referred as WE, and reference electrode, uh, RE. So this reference electrode and working electrode are connected by a voltmeter. You can see that these two are. So this is working electrode and this is reference electrode. So we apply the voltage between these electrode, okay, and we make the chemical reactions to happen. Here the redox reaction happen here, okay, and maybe the electrons sometimes may be released or consumed, and the electrons which are released here, if it undergoes oxidation, electrons are being released from the working electrode, and those electrons comes here, and we have one more electrode that is called auxiliary counter electrode. This is counter electrode. So where that electron being consumed and the counter reaction taking place. If oxidation is happening at working electrode, the reduction reaction happening at the counter electrode, and vice versa. Okay, so if the reduction reaction is happening at working electrode, then the oxidation reaction is happening at the counter electrode. And all of them are placed in a analyte solution, and we have a signal generator here. So this is based on complete electron transfer reactions at the electrode electrolyte interface. Okay, the target analyte here. So we have an analyte here. Analyte. That analyte should undergo oxidation or reduction, and that generates the measurable electrical signal. Okay, it may be the voltage change, or it may be the current change, or it may be the resistance change, or it may be the impedance change. Okay, so this sensing phenomenon is mainly governed by Faraday's laws of electrolysis and the Nernst equation. Okay, so let us understand the mechanism of this electrochemical sensing. The steps involved in this, the analyte, this is the analyte which is present in the test solution, reaches the electrode surface, maybe the working electrode. There it undergo electrochemical reaction. It may be oxidation or reduction. That depends on the nature of the substance. So as a result of that, electrons are sometimes released if it undergo oxidation, or sometimes electrons are consumed if it undergo reduction reaction. So then the charge transfer produces on the surface of the electrode. If it is current is measured, it is called amperometric sensor, electrochemical sensor. If voltage is produced, then it is called potentiometric electrochemical sensor. If there is a change in the resistance, if that is being measured, that is called conductometric electrochemical sensor. So whatever the signal that we get, the electrical signal, the intensity of that signal is directly proportional to analyte concentration. So from that, that sensor is able to quantify uh, the composition or the concentration of the analyte. You can see here, this is actually the commercial electrochemical sensors which are used for various gases, right? Various gas. You can see that there are three connections. This is because of the three electrode system. So and also the potentiostat. So as I mentioned in my previous slide, so it consists of a counter electrode, working electrode, and a reference electrode. Reference electrode and working electrode are connected by the voltage, and counter electrode and working electrode are connected by a meter, because there is a exchange of electrons between the two electrodes. So we have to measure the current. So these so voltage is input, the current is output. So in most of the electrochemical sensors, actually the inside of that, this is the inside of that, they're all consisting of three electrodes: working electrode, counter electrode, and reference electrode. Uh, and this is another kind of electrode. They're called screen printed electrode. It is also electrochemical sensor, but it is a very thin film, flexible, and it's used in various biomedical applications, and also for the glucose. For diabetic patients, they use this. In this strip, all the three electrodes like counter electrode, reference electrode. This is the reference electrode. This is the reference electrode. This is generally AgAgCl paste, and this is actually the carbon, and the working electrode is also some carbon or functionalized material. So these are the three electrodes which are actually printed on a substrate. Okay, and it is being connected to a small potentiometer, potentiostat, and from that we can get the signal. So this is the simplified form of this electrochemical sensor used in various biomedical applications. So this generally the calomel electrode is used as a reference electrode, and the working electrode we use the carbon based and coated with some nanomaterials, etc. And the counter electrode is generally the platinum electrode. Okay, so it consists of working electrode, reference electrode, counter electrode, and electrolyte. So the electrolyte solution, and it is covered with encapsulation and electrical output terminals. So it has a signal conditioning unit, potentiostat, data acquisition unit, and display controller. So these are all the components which are actually used to construct electrochemical sensors. So electrochemical sensor, it's a device that converts the chemical composition of the data of the analyte into analytically measurable signal. So as I mentioned, it consists of working electrode, counter electrode, and reference electrode, and it has a potentiostat. Potentiostat is an electronic instrument that controls the voltage between the reference and the working electrode. The reference and working electrode. So the working electrode is a very sensitive to any change in the analyte solution. It provides the surface for the analyte to undergo the reaction. So when the potential is applied between working electrode and reference electrode. The counter electrode helps in completing the circuit by allowing the reaction, which is opposite to working electrode reaction, to happen. If oxidation reaction happens at working electrode, the reduction reaction will take place, and vice versa. The reference electrode is independent of the analyte and other ions concentration. It does not change its potential. Whatever, if you put XYZ also in the analyte, it will not change. Its potential is constant while measuring the potential of working electrode. So by measuring the current of the redox reaction, the analyte can be identified and also the quantified. Based on the type of inputs and outputs, the electrochemical sensors are classified into amperometric sensors, where the output is current. So measure the current by applying the constant voltage. It's highly sensitive. Then we have a potentiometric sensor. We measure the potential difference. So there is no net current flow, and it is generally the pH sensor. So it's because of the difference in the concentration of the electrolyte, analyte solution between the electrode, it develops the potential. Then we have a third type, that is conductometric. So there is a measure, it involves the change in the conductivity, measuring the change in the conductivity that is because of the change in the resistance. All these sensors are very simple structures used in various sensing applications. So some of the materials used in with these sensors are platinum, noble metals like gold, silver as a counter electrodes, and carbon-based graphite, glassy carbon, carbon nanotubes, graphene. These are used as a working electrodes. Metal oxides like zinc oxide, TiO2, tin oxides, or enzyme. These are all used in as a working electrolytes. Some of the materials. So enzymes are used as a catalyst for most of the bio sensors. The advantages of electrochemical sensors are they're highly sensitive and good selective, and also they have very low detection limits. Even if the small amount of the analyte is present, even in ppb level, it can sense. Compact and low power, it can consume less power, and it's very compact. Suitable for liquid samples. There are some limitations of EC sensors. So the EC sensors, after using several times, that electrodes, they spoil, and they start fouling, smelling. And even the sensitivity and selectivity depends on the temperature. So therefore, it has to be maintained constant. And sometimes there is also interference effects. If X is measuring, if Y also interferes with respect to the current or resistance. And if they are made up of enzymes, so then they have a limited lifetimes. They cannot last long. Now let us discuss the glucose sensor, which is widely used. So it's the most common electrochemical biosensor used for diabetes monitoring. It's actually enzyme catalyzed, based on the enzyme catalyzed reaction. It measures the glucose concentration in the blood. Okay. So this consists of the working electrode and of course the counter electrode, and we have a reference electrode. Okay. So this is the working electrode, and you can see that, and reference electrode, uh, and this is the device which is consisting of an inlet for glucose or blood. You can say blood containing glucose. And then inside we have in the tube, it is consisting of the enzymes. Enzymes are being coated here. So enzymes. First, that blood is made to react with that enzyme, where that enzymes absorbs glucose. Okay, and the dissolved oxygen, that is SpO2. O2 is from SpO2 of the blood. So both of them, they absorb on the enzyme. So the enzymes are immobilized. Okay, so then that glucose and oxygen on the surface of the enzyme, they release H2O2. That H2O2 enters, it will go and absorb on the working electrode, and there is a polymer barrier which prevents the glucose interaction. So only H2O2 will react with working electrode. That H2O2 releases electron. So it undergoes oxidation, releases electron, and here it undergoes reduction reaction, that counter electrode. So let us look at how the glucose is being reacted with enzymes, and how it releases H2O2. So this is the beta D glucose, and you can see that this is actually OH. This is understood OH. Okay. So this is OH, and so glucose reacts with GOD, glucose oxidase dismutase. This is an enzyme which is generally used in this sensor. So glucose adsorbs on this GOD enzyme. Okay, where first it undergo oxidation to become the OH to ketone. You can see that. So a ketone is going to form. So this one. Then it undergo dehydration. There is it loses one water molecule, and as a result, it opens up that ring, and it forms gluconic acid. It forms basically alcohol into ketone, ketone to acid. So then this GOD takes out these two hydrogens, this hydrogen and this hydrogen, and it becomes GOD. Actually, this is FAD. This GOD FAD, it takes out two hydrogen from the glucose. That's why it is called oxidation. So that GOD FAD reacts with oxygen. So this hydrogen from the glucose and this oxygen from SpO2 on the surface of the enzyme, they combine to form H2O2. So H2O2 is being released, and GOFAD, it is recovered back. Okay. So again, it reacts with the glucose. Okay. Glucose. And then glucose becomes lactone, and it becomes GODFAD, becomes GODFADH2. Okay. So that hydrogen is basically taken from the glucose. So the overall reactions are at the working electrode. So the glucose oxidation takes place. The enzymes are used here. These are the immobilized enzymes here. And this is the inlet and the unreacted blood. This is actually the inlet sample, analyte sample, where glucose oxidase is actually, you can write GODFAD. I will explain the structure of this. So this is the enzyme catalyst used. So this reacts with the glucose, and it is getting converted to GOD FADH2. FADH2. And this is actually galactone. It is getting converted to ketone. This is OH is getting converted to ketone. So glucose then reacts with oxygen, becomes gluconolactone, and H2O2 is being released. Then that H2O2 enters here near to that working electrode. The first step is actually enzymatic reaction. The first step is enzymatic reaction. The enzyme catalyzes the liberation of H2O2. 1 mole of glucose liberates 1 mole of H2O2. Then that H2O2 goes near to the working electrode and undergo electrochemical oxidation. So this H2O2 releases electron and 2H plus 2O2. Then the 2H plus and O2, okay, they goes near. Oh, so here the electrons are released and the current is being produced here. Okay. So then at the counter electrode, the electrons from the working electrode are consumed and it is measured with a meter. Then the electrons are made to go near the counter electrode. This is the counter electrode, where O2 and H plus from the analyte itself, they go near to the counter electrode internally, and these electrons externally. So these electrons externally, and they form water. So they result into the form of water. So this is an overall reaction that takes place in the glucose sensing mechanism. So this is an overall reaction. Glucose plus O2 gives gluconolactone plus H2O. So this is the picture structure of how the glucose is being. This is a beta D glucose. You can see that this H and OH, okay, attached to this is GOD and FAD. This is the enzyme used. Enzyme, and this takes out these two hydrogens. These two hydrogens. You can see it becomes ketone here. So GOD FAD combines with the glucose. Glucose is getting converted to ketone, and GOD FAD is getting converted to GOD FADH2. Then GOD FADH2 plus oxygen, they will combine to release water, H2O2, hydrogen peroxide. So then this further undergo hydrolysis. Okay. So this becomes gluconic acid, COOH. This becomes gluconic acid. This is OH and H. Hydrolysis. You can see this is the structure of the enzyme. Glucose oxidase enzyme that is called GOD or GOX. So this is how it looks. This is glucose oxidase. And if it combines with this one, FAD, this is actually FAD. This is glucose FAD structure. I'll just show you the FAD. FAD is a flavin adenine dinucleotide. This is actually a very complex structure of FAD. So only for understand. That is about the mechanism of glucose sensing. Next, let us look at the ascorbic acid sensor. So electrochemical sensor is also used to sense ascorbic acid. We know ascorbic acid, it's also called vitamin C. It's an essential antioxidant biomolecule. Because it is one of the problem with this is it cannot be synthesized by human body. However, it is essential to maintain our health because the deficiency of ascorbic acid causes scurvy, mental disorders, and other health issues. So it can also be detected by using electrochemical sensor. Let us look at this. So I'm just taking this screen printed carbon electrode. Okay, it is having this is the counter electrode and reference electrode, that is the silver paste, and center the circle is called working electrode. If it is expanded, that the working electrode is actually functionalized with some nanomaterial where ascorbic acid. So this is a structure of ascorbic acid. You can see that the difference is it is hydroxy OH group. The difference in the ascorbic acid structure before and after sensing is highlighted in the blue color circle. Before it is two OH groups, and after that it is ketone group. Okay. Here the ascorbic acid diffuses into the working electrode, this one. Okay, where it undergo oxidation. Okay, so it gets converted to dihydroascorbic acid. Okay, 2H plus are being released. 2H plus 2 electrons. So this is dihydroascorbic acid. So that electrons are made to come out through working electrode and enters into counter electrode, and the counter electrode are opposite reactions that is taking place. So here the ascorbic acid undergo oxidation and releases electron. So that results in a current. So electrons are released during oxidation flow through the external circuit, and this generates the electrical current, and the current that is produced is proportional to the ascorbic acid concentration. Okay. In addition to this, that we have discussed, glucose sensor, this biosensors, glucose sensor, the what I have discussed here is the first generation sensor, because it measures the H2O2 and O2. And second generation is also being developed. They're called, they uses redox mediators. Third generation is direct electron transfer. And nowadays, since the enzymatic sensors are not lasting for long time because they have a stability issues, so people are focusing on non-enzymatic sensors. They use nanomaterial based. Actually, my research area is in this. My I'm currently working on developing a nanomaterials for non-enzymatic sensors. So there are also these electrochemical sensors are widely used in medical field for glucose monitoring, cholesterol sensing, lactate sensors, and also for environmental monitoring like water quality monitoring, heavy metal detection, and the pH measurements. And in case of industry, they use it for gas sensors, process monitoring, and corrosion sensing, etc. And widely used in wearable and IoT sensors, Internet of Things sensors, like sweat sensor, smart health monitoring, point of care diagnostics. So these are the various applications of gas sensors. Okay, this is the pictures showing the electrodes that we are using in our research lab. This is actually the platinum electrode. Platinum electrode is generally used as a counter electrode, and this is a calomel electrode. This is how it looks. Calomel is having a mercury. You can see that the mercury mercurous chloride paste is H2Cl2. So this is generally used as a reference electrode. And this is the AgAgCl. This is also used as a reference electrode. And this is a glossy carbon electrode. It's widely used as working electrode. So on the surface of this, we functionalize nanometer and we use it. The combination of all the three electrodes are being used. Sometimes we use calomel as a reference. Sometimes we use, that depends on the nature of the electrolyte. So this is the screen printed electrodes widely used in biomedical field. Uh, it is actually all the three electrodes are printed. That's why it's called printed electrode. You can see that the counter electrode, it looks like a C shape. It's made up of carbon. Working electrode is also made up of carbon in spherical shape. And this is the white one. So the white color one is actually the reference electrode. It is generally made up of AgAgCl. This one, this portion. Here the analyte drop is made to fall on that electrode, and there is a potential start to apply the voltage and then measure the current. Next, let us look at the application of electrochemical sensor in gas, for gas sensing. So electrochemical sensors, gas sensors detect toxic and combustible gases like oxygen, carbon monoxide, carbon dioxide, H2, NOx, or LPG, etc. So the mechanism here is also the redox reaction. It consists of the working electrode. So working electrode is actually sensing electrode, and the counter electrode. So this is the counter electrode. They are separated by the electrolyte. So this is some liquid electrolyte where the gas molecules they absorb on working electrode. They undergo oxidation and releases electron. They undergo oxidation, releases electron, and an ion is formed. Sometimes H plus. Then the H plus enters towards that counter electrode, or any ion which is produced at the counter electrode. They may move towards counter electrode and to go opposite reaction. So that is a mechanism involved in these gas sensors. Here the gas, target gas enters to the sensor through the capillary membrane. I'll be explaining this in detail in the next slide. So this is actually the typical representation of the how that gas sensor membrane setup consisting of. It's basically consisting of three electrode, the counter electrode here. So this is the counter electrode, this one. And then we have the next one is a reference electrode and a working electrode. You can see there's the working electrode. Next, we have a hydrophobic membrane, which is covered. Then we have a O-ring seam. Then we have a filter and the inlet. This is the capillary for the inlet. And this is an anti-condensation membrane. So, and everything is covered with a gasket. And these are the pins to connect all the three electrode, working electrode, counter electrode, and all the three electrodes are actually in contact with the electrolyte. Electrolyte is filled. Here the target gas enters. So here the gas enters through the capillary, the first step. When it enters to the capillary, then the filter removes the dust and interference. That is here. This is a filter removes the interference and the dust. Then it moves towards the anti-condensation membrane. That anti-condensation membrane actually before that, it prevents the moisture effects. So then once it filters that, once the waste, the interference are being removed, then that gas is made to pass towards the hydrophobic membrane, which allows the gas diffusion but blocks the electrolyte leakage here. Then we have a O-ring seal that supports for airtight operation, this O-ring. Then that gas reaches the working electrode. Okay, so that sensing electrode. So where gas undergo oxidation or reduction. So as a result of electrochemical reaction, the electrons are being released or consumed. As a result, the current is generated. A reference electrode maintains the constant potential as always. Counter electrode completes the circuit by allowing the opposite reactions to happen. The electrolyte enables the ionic conduction between the electrodes. So the generated current flows through the wires and pins. So the output current is directly proportional to the concentration of the gas that we are determining. So this sensor operates under non-equilibrium conditions used for continuous gas monitoring. So this is the working mechanism of the electrochemical gas sensor. It's actually amperometric. We are measuring the current. So you can see this is a typical representation of the carbon monoxide gas sensor. In this, carbon monoxide gas diffuses to the working electrode here. At the working electrode, the carbon monoxide, water, okay, and they become carbon dioxide. So yeah, so they're getting converted to carbon dioxide, and they release H plus and 2 electrons. The electron generated flows through the external circuit, okay, and the flow of electron is measured by using a meter. Then the protons generated at the working electrode, here this protons generated at the working electrode moves towards the proton exchange membrane. Through proton exchange membrane, which allows only the H plus ion, it prevents the flow of electrons. It blocks the electrons. Then at the counter electrode, okay, that H plus and the O2 plus 2 electron combine to form water. So the reduction reaction takes place. So this reaction balances the oxidation at the working electrode. So O2 enters at the counter electrode site and H2O is produced as the reaction product. So this is the mechanism that is involved in the carbon monoxide sensors. Next, another thing is it is widely used, electrochemical sensors are widely used in VOC to determine volatile organic compounds. So VOCs are carbon-based compounds that easily evaporate at room temperature. For example, acetone, alcohol, ethanol, methanol, benzene, toluene, formaldehyde. So these are generally produced from the paint and solvent and fuels industries, and also from industries, and also from the human breath. They're also called biomarkers. So these VOC sensors can detect the concentration of VOC, converting the chemical interaction into electrical signal. It's used for air quality monitoring, safety, and also the healthcare. The sensing mechanism in VOC sensor is it involves the gas adsorption on the sensor surface. This is the first step. Then the surface chemical reactions takes place, maybe oxidation reduction. Then once they undergo oxidation reduction, there will be a charge transfer between the gas molecule and the sensor. So, and the output can be electrical resistance, current, and voltage. It is being measured. So these are some of the examples of VOC, hydrocarbon-based, heptane, octane, nonane, decane, etc. Aromatic hydrocarbons like benzene, toluene, styrene. These are all carcinogenic VOCs, xylene, etc. Sulfur compounds, DMS, dimethyl sulfide, halogen compounds, ketones, alcohols, ester-based, and terpenes. So these are all volatile organic compounds. So for VOC sensors, we use tin oxide, zinc oxide, tungsten oxide, and TiO2 as a sensing material. So this sensor can operate at elevated temperature between 200 and 400 degree. Since we are using metal oxide, it becomes sensitive at very high temperature. So in this sensor, the oxygen absorbs and traps the electrons, and resistance increases. So in the presence in the VOC, VOC reacts with the trapped oxygen, and as a result of that, it releases electrons, and then resistance decreases. So when oxygen absorbs and traps electrons, so the resistance increases. When VOC enters, then VOC reacts with that oxygen, and it makes that electrons are being released back. As a result, the resistance decreases. So by measuring the change in this resistance, we can determine we can quantify or qualitatively and quantitatively we can identify that volatile organic compound. You can see that acetone reacts with the oxygen that is absorbed and getting converted to CO2 and H2O and releases electron. So this results, the released electron increases the conductivity and resistance decreases, and it produces the measurable signal. So some of the characteristics of VO sensors are they're highly sensitive and selective. They have very quick response time, quick recovery time, and you can see the detection limit is very low. Even they can go up to ppb, parts per billion level, and also the operating temperature is between 200 to 400 on long-term stability, not like enzymatic sensors. Enzymatic sensor cannot withstand high temperature because they're all biomolecule based. However, these VOC metal oxide sensors are very stable. So these VOC sensors are simple structures, highly sensitive, fast response, compact, and low cost, suitable for continuous monitoring. However, there are some limitations. They have some cross sensitivity to other gases, and they also loses their efficiency in the presence of humidity and drift over time, and high operating. Actually, they require high operating, means 200 to 400 is actually quite high. So these VO sensors are extensively used in environmental and indoor air quality monitoring, for air pollution monitoring, smart buildings, ventilation controls, and also for industrial safety like solvent leakage detection, chemical plants. This is very important in chemical industry, for the workplace safety. And very importantly, this VOC is largely used in breath analysis in medical and healthcare, and also for diabetes monitoring like acetone, disease diagnostic, etc. And they're also used in consumer and smart devices, smart home sensors, wearables, IoT based monitoring system. You can see that if you're interested to develop this VOC sensor, you can refer this journal. So this is actually accessible. Yeah. So they have developed a nanomaterial for the acetone sensor, so VOC sensors, and you can see that VOC plus oxygen which is trapped. First, oxygen traps and resistance decreases, and this is the nanomaterial. And the acetone, you can see that first once it absorbed, the acetone is getting converted to acetate, and then it undergoes oxidation to become formate and carbonate, and then it is getting converted to carbon dioxide and water. Okay, so this is a mechanism involved in acetone in VOC sensors. So for more details, you can read this article. This is only for if you are interested in PBL or EL. To do some projects in EL and PBL, you can refer this journal and make use of this. And also the another one. Okay. So this is also used for acetone VOC sensor. With this, I summarize about the electrochemical sensor and the electrochemical sensor. So in this recording so far, we have understood what is the meaning of sensor. That is a device that converts physical and chemical signals into measurable electrical output. Electrochemical sensors detect the chemical analyte via redox reactions. The signal measured here is current or voltage, or mostly amperometric, or sometimes the resistance in conductometric. It consists of working electrode, counter electrode, reference electrode, and it is also printed in the form of screen printed electrode. It's very compact and disposable. The construction and working involves, or the analyte diffuses into working electrode, where it undergo oxidation reduction reaction, and the electrons flow generates the signal. The signal intensity is directly proportional to the concentration. So we have also discussed about various materials like platinum, gold, silver, carbon, metal oxides. Enzymes are being used in electrochemical sensors as a counter electrode, also for working electrode. And we have discussed about glucose sensing mechanism, ascorbic acid sensing mechanism as a biosensor, and VOC and gas sensors, the mechanism, the construction of these devices. And we have discussed about their advantages like high sensitivity, low power consumption, and also the portable in nature. And also some limitations like interference, electrode fouling, lifetime issues, etc. And discussed about their application in healthcare, environmental monitoring, and industrial safety. So with this, I just conclude my discussion in this recording. Hope you understood the discussion about sensor and electrochemical sensor. Thank you.